point. And we'll look at that. We'll jump forward. So we've gone through all of this stuff, and I don't want to take any more time looking at that. So it's up there. We've talked about Leeuwenhoek, talked about scientific method, talked about Edward Jenner. Jenner wasn't a microbiologist, but he was, without his knowing it, he was working with a microbe, a virus. He just knew there was this condition that would arise that some people would get, and started to try to deduce what exactly the circumstances were that were involved in acquiring it. And he noticed that these milkmaids would get the cowpox pustules on their hands, but that was relatively minor, and those symptoms cleared up, and thereafter they were protected from smallpox. So he reasoned that maybe there's some sort of fundamental kind of substance present in those pustules, some sort of factor that could be extracted and given to unchallenged individuals who would then, when they were challenged with smallpox, would be able to resist it. And that's the beginning of vaccination. I apologize for the attack dogs in the background. They're barking at somebody walking down the road, and no one else is here to try and control them. So anyway, but yeah, so vaccination, and the point of all of this is that this is an outstanding example of implementation of the scientific problem. You put forward a hypothesis, that is a tentative explanation. You do some experimentation. In this case, ethically, his experiments were kind of really questionable. I mean, really questionable by today's standards. But then you get results that either confirm or falsify the hypothesis. His hypothesis was supported, and thus began the development of vaccines, and as we're now continuing to do. That was the beginning of it. Anyway, there's that. Now then, I didn't get into this last time, so there's some pretty interesting information here. So another important kind of landmark period in the development of microbiology was the recognition that the conditions under which people in a biomedical setting or laboratory setting work could promote unwanted results if those conditions were not monitored and carefully controlled. Going way back, people would have wounds and they would find that they would get infected. They didn't know what the infectious agent was. They just knew that there was something that changed. There was some infectious agent. Sometimes people would get wounds and get maggots in those wounds. That's kind of gross. I apologize for talking about that, but that would happen. And so in circumstances like that, people reasoned that, organisms that are the causes of infection or maggots that could be swarming and covering a wound, that conditions were such that those would just spontaneously begin growing, right? And that idea is known as the abiogenesis theory. Abiogenesis, which, if we translate that, means the development of life. Okay? So genesis is the creation, bio is living, the development of life without pre-existing life forms to give rise to that. So the A means without. In other words, it means spontaneous generation. Living things are rising from nothing, all right? That's what they assumed was going on. Didn't realize that if you get an infection, it actually starts with some source material. There has to be some microbe coming from somewhere else that enters that wound, is released in that wound, or positioned in that wound, or wherever, some point of entry, in order to cause infection. That was the old idea, the theory of abiogenesis. But along came a man who we're going to talk about here in just a little bit. I'll mention his name as a sort of prelude. A guy by the name of Louis Pasteur. I apologize for that, but that's Pasteur. And Pasteur's work led to the toppling, the dismantling of this idea of abiogenesis, or the spontaneous generation theory. He showed that if there's going to be contamination, say of food, or if you're making wine, if it gets contaminated with some organism that spoils it and causes it to turn to vinegar, or if there's a wound and infection sets up in that, in all of those contexts, the root cause of that would be organisms being introduced inadvertently from outside sources. Pasteur was saying that if you're going to get infected, there has to be some infectious agent. There has to have been some organism, some microbe around to have caused that. And so Pasteur's work influenced a number of other people, in particular this guy down here that we'll talk about. Joseph Lister was heavily influenced by Pasteur, but there was this recognition among several folks that when conditions are less than optimal in terms of cleanliness, when hygiene isn't something that is emphasized, and we don't take care to wash our hands, say, before we work with patients, to wash them very carefully and for a long enough period to remove infectious agents, then we run the risk of introducing those same infectious organisms into our patients. When our equipment like scalpels and forceps and clamps and so on that we're using, if those aren't clean, we run the risk of leading to inadvertent infection, which death. When we're not wearing a mask, or when our clothing is dirty, and all kinds of things could potentially be sources of these organisms that could harm patients. You run the risk of bringing about infection. These two guys here, Oliver Wendell Holmes was an American physician, and he made note of the fact that mothers who went to a hospital to give birth would commonly develop what was called childbed fever. It's called also a puerperal infection. I'll spell that word. I'll try. Let me put it in here. Puerperal, puerperal fever, puerperal infection. This is an infection that arises in the uterus and associated organs in mothers who have recently given birth. It also, whatever infectious agent causes it, and this is an umbrella term for a number of things that cause infection, so it could be bacterial or viral. Not only does that occur in the mother, but also often in the neonate, the newborn. Wendell Holmes noticed that puerperal infections were much more common in women who get hospitals. Ostensibly, the places where they should go to receive the best care were actually the places where they would get frequently dying. The mortality rate in some instances was like 50% or more. And the other option would be to give birth at home with a midwife. And there's all kinds of things about that that would be better if in fact the hospitals were not um, recognizing proper rules as far as hygiene and so forth. Giving birth in the home would be better because there would be fewer folks there that might potentially be carrying infectious agents. There's much less contamination going on. And midwives actually themselves, would implement, just as a rule, better hygiene than a lot of the doctors. So a lot of the doctors in a hospital setting in those days were men. And one of the things that sort of typified, in their eyes, a good doctor was having lots of gore and fluids and things on their clothing, as marks of having done a lot of work. It was a mark of experience. They literally left their clothing, their smocks and everything else dirty with pus and blood and other fluids. Their lab coat, if you will. And their equipment was never cleaned. And so, it stands to reason that they would probably be introducing lots of infectious agents. This man here, Ignaz Semmelweis, was an Austrian physician, and he too made the recognition that women who came to the hospital that he worked at were dying in tremendous numbers. His story actually is one that has been the basis for books, and there's actually a podcast that was recently aired on NPR. There's a program known as Radiolab, and they did a podcast about Semmelweis. Maybe I'll try to find the link to that and post that on eLearn so that if you'd like to, you could listen to it. They discussed, his recognition that something was not good. Something had gone awry. Women who came to his hospital, by the way, frequently came there as a result of the fact that they were too impoverished to be able to go anywhere else. Many of them were prostitutes, so the state would order them, to go to a facility that would specialize in overseeing births by these women. And a lot of the women, these were women that were incarcerated and they were pregnant. A lot of the women would really object to going there because they knew about how frequent infection and death were. It turns out that a lot of the doctors that were doing, the obstetrics, delivering infants, were also doing autopsies. They did autopsies at the same hospital. And so some of these doctors would do an autopsy with ungloved hands, doing dissections essentially of humans that had died from things like tuberculosis and smallpox and other illnesses, and then go right from the autopsy room into the labor and delivery area of the hospital. And so Semmelweis recognized this and said, hey, we got to clean up our act, and had people, he had a position of authority. I think he had risen in the ranks and was essentially an administrator at the hospital, as well as being a doctor himself. And he said, everybody needs to wash their hands. they use calcium hypochlorite, which is very similar to sodium hypochlorite, which it turns out is bleach. So they were using essentially a calcium-based bleach to wash their hands and their equipment. And he said you got to use clean clothing and all of that. Well, as one might have predicted, death rates plummeted. So very, very low death rates as a result of having given birth in this hospital. So the problem appeared to be fixed. But a lot of the doctors were resistant, even after, seeing the fruits of Semmelweis's policies. A lot of them resisted. They didn't want to have to be bothered with that. And so there were complaints lodged against him. And, there was some overarching medical board, I guess, over all the hospitals. And they ruled against him. And he was so incensed by this. I mean, it just, when you're right, and everybody tells you you're wrong, you're wrong, you're wrong. And they go back to this sort of barbaric practice, knowing full well that it's going to lead to death. I mean, Semmelweis just literally lost his mind over that. Placed in a mental hospital, and I think he was killed by some of the orderlies there that were in charge of watching over the folks. I think killed by somebody there. I can't remember the whole story, but yeah, it's a very tragic ending to his life. Now, I don't think Semmelweis was really all that aware of Pasteur. Pasteur's fame didn't build until a little bit later. But this guy, Joseph Lister, certainly had heard about Pasteur and was keenly interested in what he was doing. And he was a surgeon, and he implemented, he was one of the first to implement anesthesia in his lab, or in his surgical practice. If he used ether, he found that it really worked. So that was kind of the beginning of that. In those days, surgery was just absolutely barbaric because they didn't have anesthesia. So things like kidney stones and amputations and other kinds of things that would have, removal of tumors and so forth, incredibly painful. And the better surgeons actually were the ones who could do these operations really rapidly. That had to be sort of emphasized. The Lister introducing anesthesia kind of reduced the pain and the trauma from the surgery and made it so that a surgeon wouldn't have to necessarily just be lightning quick with what they were doing. But the more important introduction that he made was that of using aseptic techniques. So the A here on this word means without. Like in abiogenesis, we've got an A there that means without. Okay. Septic is derived from the word sepsis. So that refers to growth of unwanted organisms in some compartment of the body. And so techniques that are aseptic are designed to prevent that, prevent the growth of these organisms that cause infection. And there's many different things that are covered by that term. It's not just hand washing. That's one of the things that would be considered proper aseptic technique. But cleaning instruments and disposal of bed sheets and things like that people who had been sick had been laying on, or proper cleaning of those. Sutures. There were practices involving sutures that would prevent sepsis a bunch of different things. Um, one of the substances that he implemented to disinfect um, hands and workspaces and so forth was called phenol. We still use that to some extent. Um, its old name is carbolic acid. So this is an acid that is derived from uh, the distillation, I think, of some fluids that are released when coal is heated. Not to the point where it burns, but you can heat it up enough so it starts to liquefy almost. And some of the components from that can be removed and distilled, and carbolic acid, it turns out, is one of those. And it's a very good disinfectant. And they wash their hands with it. He also developed a sort of sprayer that used a spirit burner in it, which is a little alcohol burner that has a wick that comes up out of it. And there's a canister of water, and there's a canister of carbolic acid side by side. The water's heated up, and the vapor that's produced from that is used to generate a sort of spray that would draw the carbolic acid out into the air. You can look that up online if you want to, but know that he developed a sprayer, and he would actually have that fogging the area over the person that he was working on. So he had kind of this cloud of carbolic acid vapor between himself and the patient that he was working on. They would treat sutures with this stuff as well. Sutures were made out of the, uh, the intestines of cows. They called it catgut. So he'd treat the sutures with that, and ultimately, those sutures, by the way, would dissolve. So that was another one of his implementations, the dissolving suture. Um, but yeah, his practice was very clean. They were very attentive to this. And he was the toast of England thereafter, because people that they would go in for operations elsewhere, and you know be faced with mortality rates as high as 75% or so. When they went to Lister, their chances of survival were improved tremendously. Very low mortality rate, very low rate of death there. So, Lister introduced, he took some of Pasteur's ideas and applied them in a biomedical setting in England. This was in England. About the time of the end of the Civil War, and thereafter for I forget how long he lived, but he was still active, into the 1880s. So there's Lister, and there's podcasts about Lister that you can listen to. So podcasts and try to find those. So then here's the man, Louis Pasteur. All right, so now Pasteur, we talked about Leeuwenhoek before as sort of the father of microscopy. Microscopy used to study living things. Louis Pasteur could be regarded as sort of the father of microbiology. I mean, this is where microbiology really came into being, was as a result of his work. So, of course, he implemented the scientific method to deal with problems that he was challenged with. Some of the first problems that he worked on actually were posed not, through his own interest, but came from the wine industry in France, because wine's huge there. It always has been. But a lot of the winemakers were frustrated by the fact that a certain percentage of the wine that they would put up would spoil. It would turn to vinegar, maybe other substances, and would just be undrinkable. that would have to be thrown out. And so their profits were diminished by that. And so Pasteur did some investigation, and he discovered that fermentation is caused by microbes, by bacteria, and that they're operating on sugars that are in the grape juice, essentially. And as a result of their metabolism, they convert that to alcohol. He discovered that there were other bacteria that had a different metabolic approach, and that instead of producing alcohol would produce acid. So the answer to the conundrum was to eliminate those organisms, prevent them from contaminating the equipment being used to make the wine, and to emphasize the favor of the good bacteria. So all that work resulted in a deeper understanding of fermentation and foliage, and it set him on a path to looking at the possibility of microbes being the root causes of infections. And as a result of his work, he developed this idea down here, the theory of biogenesis, which says that you can't get something from nothing, that if there are living things, they come from previously living things, as I pointed out previously. Living things beget other living things. So if organisms start showing up in a broth or in a vat of wine or in somebody's body, and they weren't, didn't appear to have been there before, know that they were there. They were introduced probably, and their numbers were just so low in the beginning that you didn't realize that they were there. And once they began flourishing and growing in number, as their populations expanded. So that's the theory of biogenesis. And ultimately then that led to, when we applied that thinking to disease and what causes disease, that led to the germ theory of disease. That disease, infection, if it's something that's infectious, something that can be passed from person to person, from host to host, those kinds of diseases, as opposed to things like heart disease and diabetes and others that don't have an infectious origin, those kinds of diseases that are communicable are caused by these germs. And the term germ seems sort of antiquated and outdated, but in fact, it still is one that is acceptable. So a germ refers to any, microbe, the general term for microbe that can cause illness. And so, the theory of biogenesis, when applied specifically to the biomedical setting, is the source of this germ theory of disease. Now, he didn't offer a whole lot of ideas about how to prevent it. He just said, okay, we know what causes a lot of these diseases. So, a lot of his early work was with tuberculosis. He worked on a variety of different organisms that cause disease, and began to kind of put two and two together, put this organism with this disease, and some of that. Now, in the case of viral infections, he was stumped because viruses, of course, won't grow on bacteriological media. You have to have living things to culture them. So when he did the work to try to figure out, say, the causative agent of measles, he couldn't see anything under his microscope, and he couldn't grow anything from people who, from fluids from people who've been infected with measles. So that was frustrating for him. But he was prescient in predicting that one day they will find a germ. It wouldn't be a bacterium, but it would be something else, and that would be the cause of those infections. Of course, he was right. Ultimately, with the discovery of viruses that came sometime later, he was, I don't want to say he was vindicated, because he was never, he was never really defeated by that. I guess maybe he was. But anyway, he was frustrated by viral infections. So there were a few instances where that was a challenge. Also, we see his name all the time on cartons of milk, and containers of milk, and other kinds of foods that have been pasteurized to give them a longer shelf life. So there's this process, pasteurization, which we'll talk about sometime later, but for now know that it involves taking some food item, typically things that are liquids like milk, and I guess they even pasteurize beer and some other things like that. What you do is you heat them up, not so much that you change their flavor and the texture and appearance and all of that, but you heat them up enough to kill off most of the vegetative organisms in there. And it's not going to absolutely prevent spoilage, but it's going to hold spoilage off for longer. It's going to take a while for that to happen. So this is why a milk that's in the dairy case at Kroger or wherever you go to get it, when you get it, you'll see there's a little expiration date on there. Best used by this date. They're predicting about when the spoilage is going to catch up with you with the milk. So there's a good long time, several weeks before that happens typically. Now, if the milk wasn't pasteurized, a lot of the organisms that were in there, say from the cow's udder or from other sources, when it was hanged, would continue to flourish in that milk, and especially if it wasn't placed, in a cool setting quickly, those things would grow, and the likelihood of someone getting poisoned, food poisoning, some infection perhaps from that milk is also a possibility, that would greatly increase. So pasteurization reduced the incidence of people being harmed by the milk or other foods that they were ingesting, either by food poisoning or by infection. So we can thank Pasteur for that. So kind of a rock star. He was really, really active, did a lot of work, and pretty dominant in that time period where he was working. But later in life, he fell into poor health, and his productivity waned in 15 years of life. There was another person we'll talk about who became dominant then, and sort of kind of an adversary of Pasteur. We'll be talking about him in just a second. The other sort of titan of microbiology, if you will. This little thing here illustrates an important piece of equipment that Pasteur used to do his studies. This is called a swan-necked flask. And most flasks are made so that they're cut off like right here, and open. So if you were to put a broth, say of some chicken bones or some tendons from beef, or something that could be cooked to produce a broth that would give a lot of nutrition for microbes to use. If you were to make that broth and put it in a flask like that, that's open, with the mouth of the flask pointing straight up in the air, and let it sit at room temperature, well, within a matter of several hours, you're probably going to start to see some evidence of organisms growing in this cloudiness. What we call turbidity is kind of evidence of organisms growing in a broth-like medium. In fact, we make broths to grow bacteria in, commonly in the laboratory, and have to take great care to keep them from getting contaminated. So we sterilize them in an autoclave before we use them. And we certainly don't allow them to be exposed for too long to the air, because as Pasteur determined, germs are everywhere. The agents of contamination, the agents of infection are ubiquitous. By the way, I saw something about viruses. Viruses by far are the most common infectious agents. There are untold numbers of them out there, and they evolve all the time. And if um, you could count all of the viral particles that are here present in our world, in and on and around us, the number would be greater than all of the stars in the universe. So there are more viral particles than there are stars in the universe. And so, yeah, they are, in terms of numbers, they are the most abundant. Anyway, getting back to this. So, yeah, all these infectious and contaminating organisms are incredibly common. We said that they're ubiquitous. So what Pasteur came up with was this idea that you could take a flask and draw out a long tube of glass, sort of welded or fused to the top of the flask. And the idea is that you could heat that broth in there, heat the broth in here to kill off organisms, um, and then put that on a shelf. Now, what is prevented by this is entry of microbes. If you got little particles coming from the air, microbes, and they land in that hole there in the flask, they're going to settle into this little area right here. There's that little kind of dip right there. They're going to settle there. They're not going to be able to go up through this swan neck all the way back down into the flask. So this is a settling area right here. That he envisioned would capture dust and other particles that would be burying these germs. So yeah, this over here shows sterilized broth, and I would put the word sterilized in quotes because it's really actually not truly sterile. Boiling doesn't accomplish that. But for the most part, yeah, Pasteur was successful in doing this and not producing not seeing any growth, any turbidity. This was the usual result that he got. Now, if he broke the neck of the swan neck flask off after it had been heated, the prediction is, well, it's open now, and these particles could fall in there and contaminate this and cause growth. That's what this is showing on this side here. Well, then, that's what he saw, and so that seemed to support his hypothesis. Now, I'll just tell you, occasionally, like once out of every 100 trials where he did this, even with the intact neck, even in this scenario, he would get occasional spoiling, right? This was another thing that vexed him. What he came up with, and later was shown to be correct, is the idea that the heating by boiling was inadequate to kill some organisms. Some agents of spoilage were just too resistant to the heating. There would have to be some way to increase that heat or some other application of something that would kill those in order to prevent spoilage in there. So he basically was predicting the existence of some very resistant organisms. And it turns out, you watch the lab video lectures that I posted, talk about one genus of such organisms, the genus Bacillus, and there's another genus called Clostridium. These are both gram-positive rod-shaped organisms that can implement a strategy for survival, which is called sporulation. And when they go through that process, the end result is the production of an endospore. The bacterium, which was active at one time, goes into this endospore state and is dormant and is resistant. Boiling does not destroy endospores. We now know that. So Pasteur would be really thankful for somebody having found that out. Although maybe he wouldn't be. Maybe he'd be upset because he didn't find it out. But anyway, a lot of Pasteur's work was done using these swan neck flasks, and you can see that that was an important innovation in microbiological technique in those early days. And it just goes along with his idea of biogenesis. I mean, organisms aren't just going to arise de novo, spontaneously. Have to come from some source, and the only way that you can get that with a swan neck flask is if you break the swan neck off. Some other people who were active in microbiology, sort of overlapping with Pasteur a little bit. John Tyndall was one that indicated that he felt that dust and other particles could potentially be harbingers or vectors for microorganisms. Of course, Pasteur's work showed that, in fact, that was the case. So, yeah, keeping dust and other particulates from the air out of media that had been sterilized would keep those media, those nutrient broths, which, as I said, are just taking some meat or bones or some gristle or something like that, and cooking that and then drawing that broth off. If you treat those properly and keep dust out of them, they will commonly remain uncontaminated with that growth for long periods of time. And then Ferdinand Kohn was a German, actually botanist. Turns out that microbiology, the study of bacteria mainly during those days, what most microbiologists did was focus principally on bacteria because those were the only, for the most part. Kohn, if you were a microbiologist, microbiology, the study of bacteria, was subsumed under the heading of botany. Back in those days, you were either an animal biologist or you were a botanist or plant biologist. And in fact, as recently as, say, the late 50s, early 60s, if you opened up a biology textbook, bacteria would have been considered members of the kingdom Plantae, which includes plants. Now we know that that's kind of not right. That's really, really wrong, but that was the simplified view that a lot of people held early on. That has obviously changed very, very markedly since the introduction. We're able to find out all of these different groups and assign membership to those groups accurately. So we've got bacteria, we've got fungi, we've got animals, we've got plants and yeastins. Then, of course, we have the viruses and the prions representing non-living microbes. So it's very much more sophisticated look at the world that we have today. But back in Kohn's day, he was a microbiologist, but he would have been labeled a botanist. And he was the one that finally discovered endospores. So he was the one that kind of nailed it. After Pasteur had put forward the prediction that these things exist, Kohn took up the cause and actually finally determined that, yes, in fact, they're a reality. And the first ones that were seen were in the genus Bacillus. I don't remember which species he worked with, but that was his contribution. Then we come to this guy, okay? So Pasteur, I think, died in 1895. Koch outlived him about 15 years. Koch was the younger of the two. He actually started off as a teacher. He always had deep love for natural history, plants, animals, so forth. But later in life, he turned to the study of microbes. I think the thing that kind of pointed him in that direction was there were some cattle farmers in his region where he lived in Germany. He was a German biologist who had dealt with their cattle dying due to anthrax infection. And it really wasn't known what exactly caused this. Didn't know if it was some sort of microbe or what was going on. And Koch was the one who figured this out. He made that connection between the disease and bacterium, which he then named Bacillus anthracis, the bacillus that causes anthrax. And he determined that because it's an endospore-forming organism, that even after animals who've had anthrax have died and perished, the spores, the endospores from those bacilli would wind up in the soil. So soil itself could be a source of the organism, even long after cattle had ceased to live there. So yeah, he that became kind of his fascination, was trying to determine for infectious to try to determine for different infectious diseases what the actual infectious agent was that caused it. Kind of put those together. And he did this for a number of organisms, not just Bacillus anthracis, but bunches of other. He worked with TB and some others. And ultimately, he came up with this protocol for, finally fully establishing, which organism caused which disease, called Koch's postulates. And they're still implemented today, with a little bit of tweaking. When we apply them to viruses and some other organisms, you have to do things a little differently. But really, there has been little done to improve upon Koch's postulates. They're pretty solid, they're pretty strong. And so we'll talk about Koch's postulates and what exactly they entail. But yeah, you have to, if you suspect that a particular organism is the cause of some disease, you have to apply Koch's postulates. And once those are satisfied, this is a set of criteria that have to be satisfied. Once those are satisfied, you can be pretty confident that that is the organism that causes the disease. He also, was a strong advocate of aseptic technique. When we're culturing organisms, you have to be able to culture them free of contamination to develop pure cultures. And he was a champion of that and came up with ways of doing it. He was in his lab, I don't know if it was just him, but people who worked in his lab came up with some other innovations. One of them was the use of this stuff that we call agar. That's spelled AGAR. This is a sort of gelatin that comes from seaweed. So certain brown seaweeds, red seaweeds, when they are heated, and their fronds, their organs are heated, this sort of goo comes out of them. It causes this sort of mucilage to come out of them. And you can take that slime, that goo that comes out of the seaweed, and dry it out and powder it. And then that prolongs its shelf life. And you can then later reconstitute it, add some water, heat it up, and it'll liquefy. And then you can sterilize it by heating it in a pressure cooker or a big device known as an autoclave. We'll be talking about that. An autoclave is just a gigantic pressure cooker that allows it to increase the atmospheric pressure within the autoclave. It's like a great big steel sort of canister, like a pressure cooker, except usually they're much larger. And you can put some water in there and you heat that up under pressure. And what it does is it raises the temperature well above the standard boiling point of water, which is 100 degrees centigrade. Water on your stove will boil at 100 degrees Celsius. But in an autoclave, you can get that water's temperature to reach, 110, 115, 120, and even higher temperatures because of the pressure that's being implemented. There's this relationship between boiling point and pressure. The higher the pressure, the higher the boiling point. And so an autoclave can generate pressures well in excess of the normal boiling point of water, and that will kill endospores. So Koch was one of the first people to try to come up with ideas like that. And the medium that he began using was agar, poured into a little dish. And the dish, by the way, is called a Petri plate. Guess where those were developed? That's right, in Koch's lab by a guy by the name of Petri who worked for Robert Koch. So Petri gets credit for those, but he worked in Koch's lab. Agar was discovered, I think, by one of Koch's helpers, one of the people who worked in his lab, one of his assistants. But all of this came out of Koch's lab. It was a very active place. And by the way, we still use agar today, and we still use Petri plates. And we still really demand that we use pure culture techniques, and we still implement Koch's postulates. So a lot of what he did, and the other thing he did was he came up with ways of staining bacteria. So the stains that I talked about in the lab, video lectures like crystal violet and safranin and methylene blue and others, a lot of those were being used by Koch early on. So this is way more than 100 years ago that he was implementing this stuff. So pretty important figure and well ahead of his time. So we can thank all of these folks, but Koch, Pasteur, Lister, and I would say Leeuwenhoek are kind of the big four, of all of these folks that we've talked about. And I must, because a lot of you are probably nursing students or aspire to do that. Florence Nightingale, I'll just mention her, was an important nurse back around the time of the Civil War, I think. And she was a champion of aseptic technique. I don't know that she fully bought into the germ theory of disease, but she too made the recognition that cleanliness, hygiene and all of that was very important. So anyway, we've got to thank her too. All right, so yeah, here's Koch's postulates. Koch worked with TB, tuberculosis, is caused by a bacterium, Mycobacterium tuberculosis. This little picture here actually shows kind of what it looks like. They're little rod-shaped bacteria. By the way, it's got a weird cell wall that contains, in addition to peptidoglycan, some other stuff that makes it hard to stain. So we use a different stain technique for that. And anyway, way back when Koch was working and trying to figure out, okay, what's causing tuberculosis? What causes these tubercles to form in the lungs, these little sort of hollowed out cavitations that grow and expand through the lungs and ultimately cause a person to begin to experience bloody sputum and just horrible coughing and finally dying as a result of this. What's causing this? They also called TB consumption. So what causes that? And so he gathered some exudate from a person who was infected with TB and began looking at that material under the microscope and saw these organisms. And in every patient that he checked thereafter, he saw the same thing. So there's hint number one that, yeah, there's this thing that's growing in here that probably is a root cause, but, we've got to do more work to establish that. So you had TB, you had this organism growing in your lungs, biopsy tissue would reveal that. You didn't have TB, you wouldn't have it there, right? That was generally what you saw. So now the next step is to take some of that exudate, some of that, say, sputum or other fluids from the infected person and culture that organism, that suspected organism, in such a way that it grows by itself. You've got to grow it in pure culture. So that's step two. There's the operative term, pure culture. That means grow it up on agar or in a broth and do that in such a way that there are no other organisms. We don't want mixed cultures. We want a pure culture of just that one organism. Because what we want to do next is study that in isolation to see if it is going to cause infection if we introduce that into an unchallenged patient. Now, by Koch's time, ethics and so forth had advanced, and there was a new idea about the proper way of doing things. It would be butchery, it would be savagery, it would be totally barbaric. Introduce this putative infectious agent, the suspected infectious agent, into another human. So nobody would do that. That was unethical. Koch knew that. So instead of that, this was another one of Koch's innovations, was the implementation of non-human models, animals that could be used to model what happens in humans. So guinea pigs, right? I think white lab rats, I think one of those. I don't think it was just guinea pigs, but yeah, he worked with guinea pigs and grew those up and used those as test subjects. And so he introduced some of the suspected infectious agent to a guinea pig. And if the guinea pig develops similar symptoms and perished as a human would, then he knew that, hey, that adds a little bit more credence to the idea that this is the causative agent. That's step 3, right? So the last step, the one that would finalize this, would be to take from the organism that died or that developed an infection, you take some fluid or some tissue from that organism and to re-isolate the suspected organism. It's the same as the one that was found here. You could show that they're the same organism. Then you know, almost without a doubt. Scientists always have to reserve a little bit of doubt, but with almost no doubt, you could say that yes, Mycobacterium tuberculosis is the cause of tuberculosis. I don't think it was named Mycobacterium tuberculosis until after Koch had made this connection. Yeah, so that was one of the instances where Koch applied his postulates, was in the study of tuberculosis. So lots of innovations from him. And note his name is pronounced Koch. It looks like it would be pronounced Koch, I think is the Americanized version of that, but it's Robert Koch. And so there's that. Okay, so that's the end of the first chapter's worth of material. So I'll be starting probably tomorrow. It's now Saturday. Probably tomorrow I'll be posting the first lecture on will be chapter 3, which deals with microbiological techniques. There's some stuff in there on microscopy, more on media. There's a bunch of stuff in there, but you'll learn about the several eyes of microbiological investigations. I'll leave you hanging as to what those are. There's several things that go into a microbiological investigation, and they all begin with I. So we'll be talking about that. And so when I get through that material, I'll post that. And I think we'll probably be looking at doing our first test, probably Friday of this coming week. And so at the end of the second week, we should be able to do our first test, and that'll be over chapters 1 and 3. We're going to skip over the stuff from chapter 2 because it's all chemistry, and much of the chemistry in there is probably a significant amount of review for most of it. Not like essential that we go through that to understand later chapters. And what I find is that it winds up being a real drag on the course to get into that chapter and spend all of that time on that. So we just dispense with that. Okay? the stuff that you need to know, I'll tell you as we go along. Anyway, all right. I'll see you later.